Hello everyone. In a previous video we have studied about the characteristics of the genetic material and the historical background of the nucleic acid. In today's video let's study about the experiments which help to prove that DNA is the genetic material. One such experiment is the Griffith's experiment of transformation. The Griffith experiment of transformation was first discovered by Frederick Griffith in the year 1928. He used the bacteria Streptococcus pneumoniae, which caused a type of pneumonia in mice. And he also used mice in his experiment. So, we have two strains of the Streptococcus pneumoniae. One is the rough strain, which was not capsulated. The other strain was the smooth strain, which was capsulated. There were three strains of rough strain, R1, R2 and R3. And again three strains of smooth strain, S1, S2 and S3. He selected R2 strain in the rough variety and S3 strain in the smooth variety in his final experiment. The rough strain was non-virulent, that is, it is non-pathogenic. It did not cause any disease. Whereas the smooth strain which he used were virulent, that is, it were pathogenic. Let's recall once again. The Griffith's experiment of transformation is an experiment conducted in order to prove that DNA is the genetic material. It was first started by Frederick Griffith in the year of 1928 and he used the bacteria Streptococcus pneumoniae which caused pneumonia in mice. He also used mice in his experiment. There were two types of strains of Streptococcus pneumoniae. One is the rough strain, the other one is the smooth strain. The rough strain were non-capsulated, that is they are non-capsulated, and the smooth strain were capsulated. There were different strains of rough variety. He selected the R2 strain and here in the S strain he selected the S3 strain. The rough strain which were non-capsulated were non-pathogenic or non-virulent, that is it did not cause any disease, whereas the smooth strain which he selected were virulent and they were pathogenic. Now let's move on to the experiment. So we know that the rough strain was R2 strain which was non-capsulated and non-virulent, whereas the S strain, that is the S3 strain, were capsulated and they were virulent, that is they were pathogenic. Now let's see the four experiments what Griffiths performed in order to say that DNA is the genetic material. In the first experiment, Griffith injected the R2 strain into the mice. For no surprise, since the R2 strain was non-virulent, that is it was not pathogenic, the mice survived. In the second experiment, he injected S3 strain into the mice. Again, for the no surprise, the mice died because the S3 strain were virulent, that is pathogenic, and it was capsulated. In the third experiment, he injected the heat-killed S3 strain into the mice. The mice survived. This was because the S3 strain, the capsulated strain, was heat killed, and when it was injected into the mice, since it was non-pathogenic because the S3 strain was heat killed, the mice survived. In the fourth experiment, he combined two things. He combined the live R2 strain with the heat killed S3 strain, and he injected this mixture into the mice. For surprise, the mice died. And seeing this, he concluded that there was some material in R2 strain which was transformed to S3, and this S3 got transformed into live S3 strain. But this conclusion of Griffith could not say that DNA is the genetic material, because in his conclusion he gave a plain explanation saying that there was certain substances, certain material in live R2 which made the heat kill S3 to get transformed into live S3 and made mice to die. So this was not a perfect explanation in order to prove that DNA is the genetic material. In this video, we have studied about the Griffith's experiment of transformation, where we have stopped in the fourth experiment of the Griffith, where the R2 strain and the heat killed S3 strain were injected into the mice died, and where the Griffith gave a conclusion saying that there are certain materials in R2 which transform the S3 into live S3. But this was not a perfect conclusion. About 16 years later, three scientists gave a perfect conclusion for the fourth experiment of Griffith, which proved DNA is the genetic material. Let's study about this experiment in the next video. Until then, happy learning from H to all.